Welcome, everybody. Thank you all for attending and for listening after the meeting, to those of you who will catch up, to the fourth event in our first series. This event is entitled Addressing Barriers to Uptake, Adherence, Misinformation and Anti-Science. And I'm sure you'll all agree with me, this is even more timely during 2020 as we live through the COVID pandemic. So before we start, I wanted to give you a little bit of housekeeping. Many of you will be very familiar with this. This webinar is being recorded and live streamed via Facebook. The recording will be freely available on our website at www.phip.org. You may ask questions using the question box provided. We also have some questions for you to consider today, and we will be feeding back on those tomorrow at our live event. And please give us some feedback on how you feel this event has gone at webinars@phip.org. And we would encourage all of you to become a member of our great federation. Thank you. So a little bit about me. Many of you will know me already, but my name is Catherine Duggan. I'm the Chief Executive Officer of the International Pharmaceutical Federation, FIP. I joined the organisation in June of 2018, and I'm responsible for delivering the vision and the strategy of the organisation, working with all our volunteers and our experts and our bureaus and our excos, and of course the FIP team. So a little bit of background to the programme itself. Transforming Vaccination Regionally and Globally is our first FIP transformation outcome-based online programme of its kind, underpinned by our FIP development goals, which we launched last month on the 21st of September. The programme is sponsored by an unconditional grant and by sponsorship from Pfizer, and we are very delighted to work with them. The programme combines 3 series, all of which are interlinked. And as I mentioned, we're on the 4th episode of the first series today. In Series 1, events will focus on identifying the needs of the pharmacists and the pharmaceutical scientist workforce to deliver on vaccination transformation. That's where we are today. In Series 2, these events will discuss the actions required to deliver the needs identified during this series aligned to our development goals. So we develop a clear action plan, a clear idea of what we have to do next year against those goals. And Series 3 will focus on the member organisations across the 6 WHO regions and how they have or they will address these actions to deliver on the commitment. So, we have some questions that we would like to engage with you on. If you could help us with these, then you could jot your answers down in the question and answer box. So the questions that we have that are on our mind is what single factors or factor should be prioritised to transform pharmacy vaccination services globally and regionally? Secondly, what would be the most important achievement in terms of pharmacy vaccination services in your country in the next 5 years? Where do you see this going and what would you like it to be delivering? And what else should the FIP commitment to action outline and focus on so that we don't forget anything as this entire programme culminates in a commitment to action? We'll be taking these questions to our live session tomorrow, October the 6th, 2020, and I'll walk you through some details on that a little bit later. So here we are, colleagues, in this episode, addressing barriers to uptake, adherence, misinformation and anti-science. As I mentioned just a minute ago, this is never more topical than at the moment. We really need to collaborate very closely with each other to make sure that we have clear messages around the science of vaccination. Today, we have 2 recorded interviews with panellists with expertise in pharmaceutical sciences, where they discuss the science of vaccination, new developments in vaccination delivery, the state of the COVID vaccine research to date, medicine's adherence with a focus on vaccine hesitancy, which is a worry to all of us, and the impact of misinformation and anti-science on the uptake of life-saving vaccination programs. The interviews today are with scientists and vaccination experts. The first is Colin Pouton, Professor of Pharmaceutical Biology at the Monash Institute of Pharmaceutical Sciences, based in Australia, and Nikolai Petrovsky, Founder and Chief Exec of the Vaccine Australia company. Before I introduce the two interviewees, I want to take a moment to introduce our interviewer today, Professor Ross McKinnon. Professor Ross McKinnon will be known to many of you in the profession. He's currently the Beat Cancer Professorial Chair in the College of Medicine and Public Health, was previously a Foundation Director of the Flinders Health and Medical Research Institute and Dean. A former Flinders School of Medicine PhD graduate, he joined Flinders as Director of the Flinders Centre for Innovation in Cancer in 2011, after 16 years with the University of South Australia, stepping down from this role in 2018 to take on his current responsibilities. While Ross was at the University of South Australia, he was the inaugural director of the Sansom Institute between 2005 and 2009, and the associate head of the School of Pharmacy and Medical Sciences for research. An academic pharmacist, he has broad research interests in the pharmacology, pharmaceutical biotechnology, and molecular oncology areas. He's a co-inventor of patents relating to cancer chemoprevention through PharmaQuest and the discovery of novel anti-inflammatory compounds. He's a board member of the Therapeutic Innovation Australia. A former vice president of our federation, Ross is the current chair of our Board of Pharmaceutical Sciences. So, to our first interviewee, Colin Poughton, Professor of Pharmaceutical Biology, Theme Leader, Drug Delivery, Disposition and Dynamics at the Monash Institute of Pharmaceutical Sciences. He's based in Monash at the Parkville campus, and Colin has a particular interest in drug discovery and delivery. The use of stem cell and other similar technologies can assist with these areas. His extensive research spans 3 fields, as well as the use of pluripotent stem cell technology in disease modelling and drug discovery. Colin is also interested in the oral absorption of drugs and enhancement of bioavailability and nucleic acid therapeutics. His nucleic acid delivery expertise is being used to design the mRNA vaccines against COVID-19. I'm sure you'll all agree with me that Colin is ideally suited for our first interview this morning. So we'll play the video.
Okay, so it's my pleasure to talk to Professor Colin Poughton from the Monash Institute of Pharmaceutical Sciences. Colin is an active participant in vaccine development programs. And I thought we'd begin, Colin, by talking about the fact that the pandemic situation with COVID has really, put a lot of put a new dimension on vaccine development. So I'm very interested in your perceptions on what those impacts have been. And in particular, I'm interested in how you think perhaps the last 6 to 12 months of vaccine work around COVID has transformed or has the capacity to transform sort of further vaccine development. And perhaps to talk a little bit about what perhaps isn't right and what things we could actually sort of do to try and sort of move things forward in a positive direction post COVID, if you like, in terms of vaccination.
Yeah, it certainly is very fascinating, Ross, what's been happening. And obviously it changes week to week and difficult to keep up with the news in many respects. But I suppose one of the most interesting things about the pandemic is the discussion that's happened around how long is it going to take to get a vaccine, to get an effective vaccine. And there's been a lot of commentary around this. Some people saying, oh, it normally takes 5 to 6 years or 5+ years to develop a vaccine, and we're unlikely to get it in a short space of time. Then others have said, well, we're hoping to do it in 12 months. And there's a whole host of factors that I think have played into that. Certainly one of the factors is the new technologies, which we'll get into. But then there's all sorts of interesting factors like the enormous amount of money that's been put behind the development of these vaccines, public funding from the US, European Union, the UK, and so on, which has really accelerated the commercial programs enormously. And then, of course, there's the contagious nature of COVID-19 means that conducting a phase 3 clinical trial is much quicker than it would be if you were dealing with, say, a new flu vaccine, where you have to find enough people who are likely to catch flu, and it takes a lot longer to assemble a phase 3 clinical trial. So there's a number of things there which have really contributed to the likelihood that we'll get a vaccine really much, much quicker than perhaps would have been anticipated before the pandemic started. And I think that obviously it's surprised everyone, but it also means that it will change the way vaccines are produced in the future, I'm sure, particularly with emerging viruses such as this.
Yeah, well, I'm really interested in the fact that sort of, the speed of the vaccine development is something that is really quite noticeable, and the overlapping and staggered nature of the development programs, plus the learning, of course, I think from previous similar viruses.
Yes.
Do you think that's a model sort of, for future sort of vaccine development, this idea that we can have these overlapping staggered programs? And I guess one of the other things that's fascinating is the fact that a number of vaccine producers are committing to production facilities and things like that prior to actually having their the data to support their development programs. It's quite a radical departure, and I just sort of value your comments on that.
It is a radical departure. So to take the first point, I guess what Ross is getting at is that the phase 1 clinical trials, for example, started with these vaccines, the leading vaccines, before really any animal challenge tests had been done. Really, the only evidence supporting the phase 1 trials was that antibodies were produced in mice, but there were no challenge tests done. And so it's an illustration that the sort of linear nature of developing vaccines, which was the historical model, has been broken, so that you get into a phase 1 safety study in humans before you've done any kind of efficacy studies in animal challenges. And then you start a phase 2 study before the phase 1 study is finished, and the phase 3 study before the phase 2 study is finished. And this seems to be a model that has perhaps can be adopted for other vaccines. I don't know whether, but obviously the urgency is one of the factors that's led to this. Whether it would be possible to develop a vaccine for, say, flu, a new vaccine for flu in the same way, is not very clear to me, I must say. The urgency is definitely a factor here. And then the second point, which is around manufacturing, is very interesting too. As Ross was saying, that companies are committing to investing funds to set up processes without really knowing whether they'll have a vaccine to produce. And that is a really unusual and accelerated process, partly funded by public funding. So certainly, if you look at the US, the US agency BARDA has put billions of dollars into helping set up manufacturing facilities. And there's also some philanthropic funding that's gone in there as well, with the Gates Foundation and so on. And we're seeing that can be used by pharmaceutical companies to get processes in place which would normally take a long time to develop, and would probably be developed in a linear fashion once there's evidence that the vaccine is working. So that's quite right. I mean, all at the moment, a lot of effort's being put into production and how it's going to happen, how vaccines will be distributed without even having an effective vaccine. So it is a fascinating change.
Yeah, it certainly is. I mean, it seems to me it essentially is a new model, if you like, for vaccine development, driven very much by the urgency of the current situation. But I think it's going to be interesting to see how many of the changes that we're seeing, or how many of the new approaches, if you like, are going to remain in place for future sort of vaccine development. I guess it will depend on the urgency in each particular case. I thought perhaps what we could do next is talk a little bit about some of the new vaccines in development, and some of the particularly focused on the technologies. And I know you're particularly interested in one of the newer technologies, and I'll get you to expand on that significantly. But it seems to me that I think there's 180 vaccines in development around the world, and they seem to be a mixture of traditional sort of approaches like inactivated and live virus type vaccines. And then some of the more recently successful vaccines, such as the recombinant protein vaccines and the vector vaccines, if you like. And then some newer technologies like RNA and DNA, which really we don't have vaccines on the market from those technologies as yet. So I'm really interested in your take on what vaccines are in development, and in particular some of the newer technologies perhaps, and perhaps where you see them fitting into the overall picture.
Yes, and I guess the new technologies have certainly have been a big factor in the rapid development and the rapid push into safety studies, human safety studies, because I suppose the most prominent new technologies at the moment, the leading vaccines, are the mRNA and the adenoviral, which is essentially a DNA delivery system, the adenoviral vaccines. And the advantage they have is that faced with a pandemic and a new virus, once the genome is produced and once the genome is sequenced and published, then it doesn't take long to get to a vaccine product that can be tried, at least in the early animal studies, the early studies in mice to find out if you can generate antibodies. And so if you look at what happened after the first publication of the SARS-CoV-2 genome, it really only took 2 months for the mRNA vaccine from Moderna to go from genome sequence through some sort of preclinical development into a phase 1 safety study in man. So 2 months is an incredibly short time from literally finding out sequences that you could use to actually getting into humans. I don't think anyone would have believed it could have been done that quickly. Certainly it's possible to produce candidate vaccines in a short period of time, but then the confidence to go from early preclinical studies into man is a very unprecedented change. So if we look at the kind of technologies that are available now, what's different about them is that making the product and purifying it in such a way that you think it's a candidate vaccine for early stage preclinical testing is very quick. So the mRNA vaccines and the adenoviral vaccines were very much more rapid even than the subunit protein type vaccines. But even the subunit protein vaccines were relatively fast because now the technology for making proteins and purifying them is really very common knowledge, and in a sense, that process can be accelerated perhaps more quickly than it has been in the past. The other more traditional approaches, inactivated vaccines and so on, actually present much more pharmaceutical challenges in terms of purification and characterization. And that's probably why those vaccines are sort of lagging behind in terms of in the timeline. But of course, we don't know which of these vaccines will be most effective. And so that'll be the next fascinating period of time when we see how these different approaches, the new approaches, compare with the older and more traditional approaches. I suppose my take on it is that at the moment, there's no reason to think that the new approaches won't work. And certainly the new approaches from a pharmaceutical perspective are cleaner and tidier and easier to manage, easier to manufacture, easier to, if you like, do quality assurance. And so from that point of view, these approaches really haven't had an opportunity before. I guess one of the things about vaccines is that if you have a working vaccine, like for example, the measles, mumps, rubella vaccine, you don't really, it's not a commercially viable option to set about changing that and trying an mRNA vaccine for that kind of disease, or those diseases. We have a working MMR vaccine, and it's not a commercial prospect to actually develop a new one. And so the pandemic has sort of given opportunities to the new approaches to vaccination, which probably wouldn't have been there had the pandemic not arrived. So for those companies involved in mRNA, for example, it's actually an amazing opportunity for them to showcase the effectiveness. Now, whether it works is we'll have to wait and see, but certainly it's an opportunity that wasn't there before the pandemic came along. And those companies developing mRNA vaccines were really working on a relatively, what should we say, rare and less important diseases than this COVID-19. And they didn't have the opportunity to push into an area which was really of global significance. And that's, so that's
so I was just going to say, I mean, I think the really interesting sort of issue you've raised there is, in terms of transforming vaccination. I mean, these new technologies do offer clearly relatively easily assembled infrastructure. As you said, less pharmaceutical complexity, perhaps, than sort of some of the previously successful models. And that's probably one of the exciting things that's emerged from all the momentum that's been built around the sort of the pandemic. I'm really interested in your observation that your optimism in terms of sort of the efficacy of these vaccines, because as we know, we don't have successful vaccines produced by a number of these new technologies as yet. I'm just wondering if you think we've learnt anything already from the clinical programs or anything like that around, obviously both immunogenicity is a key to efficacy, but also tolerability is obviously the other key component of a successful vaccine. Do you think, how do you think things are emerging in terms of sort of the development programs of some of these different methodologies?
Yeah, that's a very interesting area, isn't it? I suppose one of the things to talk about first in that respect is that thus far with COVID-19, all the vaccines are aimed at producing antibodies against the spike protein. That has been, and that is the first objective.
So it's probably worth pointing out there that we know a lot about sort of that spike protein from similar viruses that emerged earlier. So to some extent, some of the preclinical data is available already, and that's one of the things that's accelerated the programs.
Yes, undoubtedly, yeah. So the SARS-CoV-2 virus, which is the COVID-19 virus, is very similar to the SARS virus. The spike protein that we hear a lot about, which is the target of most of the vaccines, is structurally very similar between the two viruses. And so that certainly had a major influence on the speed with which the early vaccines could go into the clinic, because the structural biology of the spike was already understood. Pretty much as soon as the genome was sequenced, you could sort of see the homology between the two viruses, and therefore we could understand what the spike looked like, and also know exactly what's the receptor binding domain. At least the expectation was that we understood it. And it didn't take long to confirm that, yes, the structure was pretty much as expected, and the subtle differences between SARS-CoV-2 and SARS really were anticipated pretty much straight away, and so they proved to be the case. And so that meant that people could essentially produce vaccines in an analogous manner to the candidate vaccines that had been developed for SARS. What happened to the SARS vaccines is that they didn't really ever make it into, they were tested in the clinic, but they didn't become approved products, largely because the SARS problem sort of went away it went away sooner than products could be developed. Largely probably because of the funding behind them. So another thing about this pandemic is because it's so contagious and spread so quickly, that funding was provided to take these vaccines forward. And once you get into phase 2 and phase 3 clinical trial, you do need substantial amounts of money to actually do the studies. And so that money was provided largely from public funds. And that really wasn't the case with SARS or MERS. So I think, you know, the learnings from SARS and MERS were definitely hugely important.
Can I ask you a question about sort of the vast majority of the vaccines under development? I think virtually all of them would be intramuscular in terms of their sort of administration. And there is some, as I understand, some evidence that the immune response to the virus in the upper respiratory tract is different to the immune response in the lower respiratory tract, and the fact that what a lot of these vaccines may achieve is actually sort of prevention or attenuation of disease, sort of rather than perhaps sterilization. Any comments in relation to that, in terms of sort of the likely efficacy of these vaccines as we roll them out?
Yeah, that's right. So I suppose that's what we didn't quite get onto in the previous question. In that the first objective is to get some antibodies against the spike to try to prevent infection. But that may not be enough to really give sustained immunity against the virus. And it may not even be enough to really help clear the virus. So this is one of the unknowns that we can tell from the preclinical studies and the comparison between preclinical studies and the early safety studies in man. We can tell that antibodies are being raised, and antibodies are being raised in the human studies just as they were in the mouse studies, and that gives us some confidence that we're on the right track. But doesn't mean to say that the vaccine will be adequately successful in the long run. And that's because it may well be that cell-mediated responses are needed to really give sustained immunity against the viral infection. And at the moment, it's unknown the contribution, the relative contributions of an antibody response versus a cell-mediated response, and the relative contributions of the response against the spike protein versus all the other proteins in the viral infection as well. And so that's the unknown, I think, in terms of efficacy. We're trying to, at the moment, to correlate antibody production with efficacy, but what we don't know is whether all the other components are a really important part of immunity against the viral infection. And you know, as you point out, Ross, that the need to clear the virus may be different depending on its initial exposure versus the sort of chronic nature of an infection which has now gone to other organs, into the deep lung or into the cardiovascular system and so on. And I think perhaps one of the most interesting parts of this whole exercise at the moment is this issue about whether there are good and bad antibodies. So, and the issue
What's going to be my next question?
So, well, just to give the preamble then, I guess, with the SARS and MERS infections, we understood that some individuals raise antibodies against the spike proteins of those viruses, which are not particularly useful, in fact, can potentially be damaging. And there's two aspects to this. One is that whereas we work on the assumption that in order to neutralize the virus and prevent infection, we have to raise antibodies against the receptor binding domain. And the receptor binding domain is a small fraction of the whole spike protein. So there's one issue, which is, well, we can raise antibodies against the spike protein, but what if those antibodies don't actually bind to the receptor binding domain, but bind to other epitopes in the spike protein? So those antibodies are normally referred to as non-neutralizing, because in vitro tests and in tests for, say, viral infection of cells in cell culture, they tend not to prevent infection. They tend to bind to the spike protein, but they're not necessarily very effective in preventing infection. Now, if you vaccinate with the whole spike protein, you might expect to get antibodies which do bind to the receptor binding domain, but also those that don't. So-called neutralizing, but also non-neutralizing antibodies. So the question then is, well, are the non-neutralizing antibodies bad? Or are they just not very useful? And that's an area which is not totally understood with SARS-CoV-2. And the reason I say that is that with MERS in particular, there are some reports that some of the non-neutralizing antibodies could actually make infection more likely. So so-called antibody enhancement of infection. Now, we don't know whether that happens with COVID-19. We know that it happened with MERS, and to some extent the thinking was that it also happened with SARS, but it may be that the SARS-CoV-2 vaccine is already, the SARS-CoV-2 virus rather, has very high affinity for its receptor, the ACE2 molecule, and it could be that really it's already an extremely strong binder and it can't be enhanced. But there was some evidence with SARS and MERS that the binding could be enhanced by these non-neutralizing antibodies. So that's one aspect that is a little concerning. Another aspect is that what happens when a patient sees the virus and the virus gets coated with these non-neutralizing antibodies? So when I talk to virologists at the Doherty Institute and collaborating with, one of the things that they think is that if the virus gets coated with non-neutralizing antibodies, then the virus will tend to be taken up by phagocytic cells like macrophages locally, and those macrophages can migrate to other tissues, and that may be one of the reasons why some people have a really awful disease with COVID-19, because the particles are taken up and transported to other organs, and that's how they reach the deep lung or maybe the cardiovascular system. And once in those areas, the virus hasn't actually been neutralized and can leak out and cause further infection, because those organs certainly have the receptor for the virus. So if the virus if you haven't prevented infection and there's still virus around, that's how it could potentially be transported to other organs. So all of these things are unknown.
Challenges, yeah. Perhaps I could just move on to some of the other challenges. Obviously, the challenges relating to efficacy are, potential deal breakers in terms of the success of a vaccine program. One of the other things is, of course, the success of a vaccine is dependent on successful rollout of the vaccine. And, if we are talking about a vaccine that needs at least 2 doses, it's starting to look like it's going to be a multiple dose vaccine. That's 16 billion doses required to scale up for the world population. I think one of the interesting things is that many of the vaccines that are in development by companies that have not actually, produced a vaccine previously, certainly not on the scale that we're sort of talking about. So do you think there are likely to be problems in terms of scale up and in terms of sort of bottlenecks, something as simple as syringe availability or something like that? How do you think the rollout might sort of proceed?
It's a massive challenge, isn't it? Certainly, and as you say, a lot of the vaccines, the mRNA vaccines, for example, have really only been produced in in batches large enough to do small clinical trials. So to roll that out, even for a phase 3 clinical trial, and certainly then to the general population, is going to be a huge challenge. And it's unfamiliar technology to most companies, as you point out, Ross. And at the moment, the smaller companies, the innovator companies, if you like, who are behind the mRNA vaccines, are needing to set up relationships with companies that have manufacturing skills and experience, but not in this area. And they're certainly having to learn, and they're going to have to learn how to do it, and they have to build plants to do it. And I think all of those, at that scale, it's going to be a massive challenge for those companies. And obviously, what is happening at the moment, what we're seeing at the moment, is that the innovative companies that are in the process of doing phase 3 studies are really trying to find as many potential manufacturers as they possibly can across the globe, and doing deals with companies on the basis that they may be required to manufacture. So, which is fine if you know how to do it, but if you don't know how to do it, then you're making a deal on the basis that you will work out how to do it. And it's fascinating, and certainly a big challenge, absolutely. And I don't think we'll really know until it starts. And then you get onto those things, as you've mentioned, vials, syringes, supplies of those, which is the other aspect of it on a large scale. I don't think anyone really knows how we're going to cope with it in the end.
No, I think that's a really valid sort of point that you've made. I mean, essentially what the pandemic has done is it's, as you said, it's provided opportunities, and in terms of sort of advancing new technologies, certainly sort of provided the scope to transform the development of vaccines. But it's probably also thrown up a massive number of challenges that I think, some of those, as I said, will resolve themselves through phase 3 trials, but many others are things that we really don't know the answer to at this stage. I think we'll wrap up there, Colin, and I think you provided some sort of excellent insights into vaccine development through the COVID program and what the implications of that are for broader vaccination. So thank you for your time, and good luck with the development of your technology. Thank you.
Thank you very much, Ross. It's been a pleasure. It'll be fascinating to observe as we go ahead.
Certainly, yeah, lessons to be learned. Thanks, Colin.
Thank you so much to Ross and Colin there. I'm sure colleagues who have been watching that absolutely agree with me that we're probably going to need Colin back in the next couple of months as these advances progress. Such wisdom and such good experiences there. So now for our second interview. I have already introduced Ross, and you will all be aware of his eminent CV. So next I'd like to introduce Nikolai Petrovsky. Nikolai is a Director of Endocrinology at Flinders Medical Centre, with a co-joint position as Professor of Medicines at Flinders University. He is also a Vice President and the Secretary General of the International Immunomics Society. Nikolai is active in diabetes, endocrinology, and research into vaccines, and is the founder of Vaccine, a company funded by US National Institutes of Health to develop novel vaccine technologies. In 2009, Vaccine, the company, won the AMP Innovation Award at Telstra Business Awards and Australia's Coolest Company Award from Australian Anthill magazine. Nikolai has developed vaccines against influenza, hepatitis B, sting allergy, malaria, Japanese encephalitis, rabies, and HIV. He's authored more than 90 papers and chapters and is a regular invited speaker at international vaccine conferences, an ideal person for Ross to interview. So, our next video.
Okay, it's my pleasure today to talk to Professor Nikolai Petrovsky, who is the leader of Vaccine, a company that he founded, I think, in 2002. And Nikolai has been an active player in the vaccine area for nearly 2 decades now, and is particularly active around the COVID vaccine development. Thank you, Nikolai, for being with us. What I thought I'd do to start is just to ask you a little bit about vaccine and about what vaccine has done prior to perhaps COVID, and we'll get on to COVID shortly. But if you could just tell me a bit about the background of vaccine, what they do, projects they've been involved with, and so on.
So, Ross, I did a PhD in immunology alongside my medical degree in Melbourne some 25 or so years ago. And after that, sort of launched and set up my own lab in Canberra and got interested in vaccines and particularly in vaccine adjuvants. And met a chap called Peter Cooper who'd retired, but had been at the ANU, and he'd found this interesting sugar-based compound that had some quite unique immunological properties. And so we sort of joined up together and proved that this was working as an adjuvant, and then were able to show that this was able to enhance a number of different vaccines. I think we're working on mainly flu and hepatitis B back then. We set up the company around that 20 years ago to take this forward with a bit of money from Aus Industry. We then got a large contract with the National Institutes of Health in the United States, because this was just after 9/11. And George Bush at the time realized actually the US had fallen way behind in terms of the vaccine industry. It had almost ceased to exist in the US, because vaccines were not seen as sexy or profitable. And so all the manufacturing in the US had been shut down, and it had moved elsewhere to India and China. And obviously after the anthrax attacks, he realized that, the US was very insecure, having no vaccine manufacturing capability and also having no vaccine research capability. So he put aside several $1 billion to rebuild that. And we were fortunate to be one of the first players, even though we weren't from the US, to actually get a contract out of that biodefense money, and obviously have been supported by them ever since.
Right. Can I begin by sort of taking you back, obviously, just before the pandemic, and to talk about sort of, if you like, how do you think the scientific community, the biopharma sector, was positioned prior to the pandemic in terms of vaccine development? You've talked about obviously some deficiencies some time ago. How do you think we were positioned in terms of science and technology when this all happened?
So unfortunately, pandemics occur pretty frequently, really, maybe every 5 to 10 years. And during each pandemic, we always say, oh, wow, we did really badly. We've got to do better next time. And everyone gets excited, and there's lots of, committees set up. And then after a year or two, the pandemic goes away, and governments lose interest. The money dries up, and so we find, inevitably, having been now through multiple pandemics, I'd have to say we were no better set up for this one than we were for the last one, and that's really an indictment on. And it's not just a local problem in Australia. This is a global problem, that it's really hard to motivate governments to invest in pandemic preparedness, except during a pandemic, when in fact it's not a good time doing research, I can tell you. During a pandemic, it's a nightmare. It needs to be done, before or between pandemics, and unfortunately, that doesn't happen.
You're one of 100, or you're in leading a program with one of 180 vaccines that I believe are now in development around COVID-19. Could you perhaps give us, before I talk to you about your particular project, could you perhaps give us a little bit of oversight of what technologies are actually driving this frantic activity in relation to vaccine development. And whether it's a mixture of newer technologies or whether it's sort of the existing vaccine technologies that we've had obviously for quite some time, just to give us a flavour of what's actually happening from a technological sense.
So I guess this pandemic, it's been quite interesting to see the diversity of platforms that are being used. In previous pandemics, a lot of which have been influenza related or pandemic threats, because some of them still haven't turned into a pandemic, like H5 influenza and H7 influenza, but may in the future. Obviously, we had an H1N1 influenza pandemic in 2009, and there we saw the only vaccines that were developed were very traditional technologies. And typically that's because, in a pandemic, you haven't got time to explore new technologies and to go through all the issues of are they safe, are they not. So this really is the first pandemic where, for serendipitous reasons, I think, that most of the early vaccine candidates were actually the most novel, the most experimental. And as I say, that normally would never happen. And it really was serendipity for this, in this occasion, that we had a couple of novel viral vector vaccines like the chimpanzee adenovirus vector, come out of Oxford. We had the nucleic acid vaccine, so the mRNA, I guess with Moderna being one of the leaders, but now Pfizer and Merck and all the other big companies sort of jumping on that bandwagon. The DNA vaccines, which have been around a long time but have never got anywhere, and of course, so they get pulled out of the closet. And then, in fact, the traditional approaches, which are the recombinant proteins and viral-like particles, which would normally be the mainstay, have ended up being sort of towards the back of the pack. And so it's been interesting to see these highly sort of risky experimental vaccines being seen as the lead candidates because they were rolled out first. And again, that was for serendipitous reasons.
Do you think it probably also reflects
technologies behind? Sorry?
I was just going to say, do you think it probably also reflects that they're perhaps pharmaceutically simpler in terms of getting a product that is suitable for clinical trials compared to perhaps some of the recombinant techniques, for example?
Yeah, so I guess there were two factors driving it. First, that there had been recently in the last 2 years a large amount of money put out by an organisation called CEPI, or Coalition of Epidemic Preparedness Initiative, for new technologies to apply to the MERS coronavirus, which has been considered a problem but never turned into a pandemic. So those programs had just been funded in the last 12 months leading into this for, MERS, which is a coronavirus. And so not surprisingly, CEPI threw a lot of resources at those projects in the first few weeks of COVID starting, because those programs are already there, already being funded, already had sort of platforms that were being tested, and in some cases had just started human phase 1 trials, and so they ended up actually being the things that were available quickest. As well as that, you are right that some of those technologies are much simpler and faster, at least in the initial stages, to get product made in order to get it into humans. Whereas recombinant proteins are extremely complex for a whole number of reasons. And so, yes, so because they already had funding, because they were there, because they had platforms that were being applied to MERS coronavirus, and because they were quicker and simpler, or appeared to be, because I can come back to where are the catches in that.
Yes.
They certainly got the early lead and got all the attention, and with attention comes more money, and with more money comes faster progress, comes more attention. And so that really has driven those initial candidates, forward at a fast rate because, they have the resources behind them.
So you've already highlighted that some of these novel strategies or new technologies were sort of the first ones sort of into early clinical trials, if you like. But of course, as we know, generally they haven't produced successful vaccine programs in the past, whereas obviously the traditional approaches have, and certainly the approach that you're using, the recombinant protein approach, has been successful more recently with vaccines. Does that give you confidence in terms of sort of the approach you're using, in terms of where you might sort of end up in the scheme of things?
Yeah, so we sort of actually did develop a couple of nucleic acid vaccines, right back at the beginning in January. We made an mRNA and a DNA vaccine. So we didn't just make the platform because it was faster to make those anyway. So we thought that would be useful, at least to get some initial feel for what might work. But ultimately, when we reflected on, well, what are we going to put all our energy into, it was really the tortoise and the hare situation where we said, yes, we could push these new technologies forward and get them moving fast. But then when you think about it, there's no proven, as you say, pathway. There's no existing human safety data. There could be all sorts of pitfalls. How long will they work for? I mean, will they work? But if they do, how long will they work for? We don't know because we haven't seen them in humans before. So ultimately we said, look, we know we're going to take an extra 3 months to make a recombinant protein and get that to the clinic. But, ultimately, if we get that protein to the clinic and it works, well, then we know, recombinant proteins give long-term protection. We know they're incredibly safe. We know we can make them at massive scales because that's already being done. So from that point of view, we did see it as really the tortoise situation where it was going to start slower because it's harder at the beginning, but we figured that it would finish much faster because regulators particularly would be a lot more comfortable approving a traditional protein-based vaccine rather than a completely novel technology where you really can't predict the safety, early on. You need a lot of data.
Yeah, well, we've touched on a few things that I want to come back to there in terms of scale up and rollout and things like that. But perhaps this is a good moment to talk specifically about your work and what vaccine is actually developing and where you're at in your trials and so forth.
So for the last 16 years or so, we've been using a platform that involves production of proteins in insect cells. And this is considered in the vaccine world, which is very conservative, a relatively new technology. But, obviously, researchers have been using insect cells to make proteins in their labs now for 30, 40 years. And in fact, Cervarix, which is Glaxo's papillomavirus vaccine, which is given to young girls around the world, a lot of people probably don't know, but it's actually made in the same insect cell line that we're using for our vaccines. So again, this is a relatively well-established mechanism of making recombinant proteins. It offers a number of benefits, mainly because insect cells are very different to humans and to mammalian cells. So, for instance, if you have a virus in an insect cell, it can't infect mammals and it can't infect humans. Not that obviously we establish our cell lines are free of viruses, but even if there was one there we weren't aware of, it wouldn't pose a threat to humans if there was carryover. It's also, insect cells are a lot more robust than mammalian cells. They're cheaper to manufacture. So we've been working with vaccines based around this insect cell platform, as I say, for 16 years. In 2009, in fact, in partnership with a US company called Protein Sciences, we used this to develop the world's first swine flu vaccine, and we had it in trials here in Flinders under 3 months from discovery of the swine flu virus, which at that point in time was the fastest ever, human vaccine development project. And we got incredibly good results from it. We got very high levels of antibodies, and no one in the trial actually ended up getting infected with the swine flu. We also used it for SARS coronavirus vaccine back in 2004. There we tested it out, showed that it was very protective in mice, against challenge with the virus. But in fact, the funding, again, because SARS sort of ended up being controlled and disappearing, then in fact the funding ran out, so we didn't get that into humans. We used it for a MERS coronavirus vaccine, which was actually shown to protect alpacas and camels, which is a quite unusual challenge model, but obviously they're very susceptible to MERS. And we showed that we could protect them with this approach. And so, not surprisingly, having used it for so many other problems in the past, we've applied the same approach here to COVID, where we're expressing the spike protein from the COVID virus in the insect cells. We then formulate it with our adjuvant, which is a polysaccharide, plant-based polysaccharide, and then give it as a standard intramuscular injection. So we've shown that that's working in monkeys and ferrets, which are two of the better animal models for COVID. Although, to be honest, apart from monkeys, really, there isn't a good animal model. Most animals just clear the virus and don't get sick with it. Whereas monkeys, at least, they get sick, which hopefully, if you can protect them, means that your vaccine may in fact work in humans. And obviously, we've just completed a small human trial where the data is still being analysed. So we're moving forward, but we still have some challenges ahead.
Yeah, I'm sure. Obviously, as I said earlier, you're one of 180 vaccines in development, and we've heard from many people that we may need certainly more than one vaccine, and we certainly may need a variety of approaches. Can I just get your take, if you like, on the current status of the clinical sort of trials that have been undertaken so far in terms of immunogenicity, tolerability, things like that, and where you sort of think your technology might sit, perhaps in the scheme of things, in terms of the recombinant approach, in terms of immunogenicity against some of these new approaches based on the data that's emerged. Same with regard to tolerability. Just a general take on what you've seen so far.
Well, I think, the first thing we have to realise, people talk of, 160, 200 candidates, but at this point in time, there are only about 40 that are in human trials. I suspect, there won't be too many more come into trials, mainly because, again, the impetus to do that reduces the ability to get funding. If you've got a candidate that's still preclinical, basically it's going to get very difficult. We were 16th in the world into clinical trials, so at least it puts us, halfway in the pack, not in that first cohort, but, I think we were the second recombinant protein-based vaccine in the world to enter human trials, or second or third. So we're still in a very select group there, which is important because, as I say, once you have those first 2 or 3 candidates in each category that have been sort of selected and are moving forward, the others find it harder and harder to get resources. So the race actually gets harder if you're at the back. So I don't think that 180 candidates are going to hit the clinic anytime soon. I think we've really seen most of the ones that are already. In terms of differences, obviously, from our perspective, being a clinician, safety is what drives prophylactic vaccines. And a lot of people don't appreciate that because they don't understand that, if you have a side effect, saying, it occurs only in 1 in 1,000 people. If you were only going to inject 1,000 people, with cancer and one got a side effect, you'd say, look, that's an acceptable risk. The issue here is you're proposing to give your vaccine to billions of people. So a side effect, even if it's, you know, a rare side effect of 1 in 1,000, now you're getting hundreds of thousands of people getting that side effect caused by the vaccine, and that becomes a massive issue. And so you can't overemphasize safety and tolerability in a prophylactic vaccine. Very different to a therapeutic drug. So essentially, our vaccines can't be too safe, if I can put it that way. And of course, that really then starts to distinguish a lot of the different platforms, because if we ask about nucleic acids or we ask about, you know, chimpanzee adenoviruses, we simply can't answer the question. We just don't know if they're going to be safe at all, or if they are going to get a side effect, what's going to be the frequency? Is it going to be 1 in 1,000, 1 in 10,000? So that is really going to be a big issue, I think, as we go forward. And we've already seen, you know, AstraZeneca's vaccine go into clinical hold twice because of side effects that were neurologically related. And you know, that vaccine in the United States is still on clinical hold, with the FDA doing more investigation, even though the trial has been restarted in the United Kingdom and a number of other countries. So I think these safety issues are going to become more and more prominent as we're getting towards the other end, the phase 3 trials, because that's where you start to see issues, I guess. So again, I think that, you know, a lot of the vaccines may never see the light of day because they hit a safety barrier. The other thing, of course, is efficacy. So, you know, we still don't know, will one platform work better than others? Will they all work? Will they all not work? As of today, we still don't know, you know, what's going to happen in terms of efficacy of any of the candidates.
You've already touched on the critical issue that, you know, this strategy obviously has to be directed towards billions of people, if we're talking about a global pandemic, which we are. And you've already talked about the fact that from a manufacturing perspective, some of the technologies may have distinct advantages over some of the others in terms of large-scale manufacturing. But there are obviously many other barriers to uptake of vaccines, you know, and they can be based on people, they can be based on providers, they can be based on obviously system barriers and so forth. Do you think the public nature of the COVID sort of experience is likely to have positive or negative effects on uptake of a vaccine on a sort of a global scale?
Well, that, you know,
it's a very difficult question, I know.
It's a very important question. And, you know, what I guess some of us feel that, unfortunately, because in some ways the most risky vaccines are currently the highest profile, again, because of serendipity in what went to the clinic first. The big concern is if something goes wrong, and remember, they're very high risk because they've never been tested before. If something goes wrong, and that's very public, which it will be because COVID is the only thing you see on the news these days, then that will create an enormous potential backlash against not just, you know, the particular platform, but against all COVID vaccines, and potentially spill over into all vaccines generally. You know, if there was a loss of confidence of the public, you know, because something went wrong in one of these programs, I mean, that would have dreadful consequences that would flow on for decades. And so some of us, that's why we have been concerned that so much attention has been put on potentially the, you know, I wouldn't say the riskiest, but the most unproven technologies. You know, it would be better if they were developed quietly till they got to a point where you could roll them out. In this case, they're front of mind. You know, they're on all the news, and if something goes wrong, we're all going to know about it, and that will have consequences, as I say, for the rest of our lifetimes in terms of, you know, if people lose faith in vaccines, we're in a very difficult space.
Thanks for that. My last question is one that probably you could expect to come from a pharmaceutical sort of scientist or someone with a pharmaceutical background, and that is around the development pathway for vaccines. I mean, what we've seen here is a very rapid acceleration of a development pathway. As we know, normally the time frame of vaccines is much longer than what appears to be sort of happening now. We've seen sort of staggered, overlapping development programs where phases of development are being combined. We're drawing on previous preclinical data from other sort of areas and so on. Do you think this sort of accelerated model for vaccine development is likely to be with us to stay?
Well, I hope the best aspects of it do stay, because one of the frustrations with the traditional process is it's been taking 15 to 20 years to develop a new vaccine, and around about $2.5 billion, which makes it extremely difficult to get new vaccines. And so there are aspects certainly of this accelerated programs which have shown that if done right, you potentially can do it faster and potentially thereby do it cheaper, because time is money. And so, but of course, as I say, against that, we have to think, well, why did it previously take 15 years to develop a vaccine? Was that because that was mainly about safety and getting it right? And so if we try to compress all programs into a year or two, are we actually going to ultimately compromise too much? So I'm hoping that there's somewhere in the middle, that we can identify what parts of vaccine development, and you mentioned some of the preclinical testing, which, to be honest, is an anachronism. Anyone will tell you, well, the preclinical sort of safety studies that you're forced to do at great expense tell you really nothing. They don't predict safety in humans, so why do we do them? So I think that there are things there that could be removed from the sort of modern paradigm of developing vaccines, but I don't think we should compromise on testing safety, which means we're still going to need large phase 3 clinical trials, and they are the most expensive bit of the equation. So, and I don't think we can bypass that bit of it, unfortunately.
Can I just slightly extend the question as well? I mean, we're talking about prophylactic vaccines, obviously, but do you think the field of therapeutic vaccines is likely to be a beneficiary of the intense attention around the sort of the pandemic?
Yeah, look, I mean, we're doing some work in therapeutic cancer vaccines, also in the area of therapeutic allergy vaccines. And I think, yes, absolutely, that all of these new technologies that are sort of being pushed into the clinic will generate a lot of data, both on efficacy, but also on safety. And so I wouldn't be surprised at all to see some of those technologies repurposed and come into the therapeutic field, where in fact the risk benefit is very different. So even if a vaccine proved to be unsafe for COVID, say a nucleic acid vaccine, ironically, it may prove that it's safe enough to be used as, say, a cancer vaccine, because the side effects are 1 in 10,000, which may be unacceptable for COVID, but may be incredibly acceptable for cancer, where we know chemotherapy gives horrific side effects. So it's a little bit unusual, but I think there will be potential discoveries during this COVID sort of program where these technologies may find even better use in a therapeutic vaccine context. So that would be a major positive, I guess, outcome of this. But hopefully the most positive outcome will be, very successful COVID vaccines. And I'm very confident that's going to happen. I mean, the animal data, I think, does suggest that we should be able to prevent COVID with a good vaccine.
Okay, I'll leave it there, Nikolai. I want to thank you for very deep insights, obviously, into vaccine development and the state of play at the moment. Really appreciate your input. Thank you very much.
So, colleagues, I'm sure that you will agree with me that Nikolai and Ross had a very wide-ranging conversation. One of the things I wanted to point out to you all was the emphasis on safety that Nikolai went into, and that patient safety is one of our 21 FIP development goals. Episode 7 of our second series will focus on patient safety. That's planned for the 18th of November. And I note that Lena has already put a link to the details of the events on the chat box, but we'd really love you to engage and participate and tell us what's needed in your setting and your country for the FIP commitment. And in doing so, I would compel you all to join us at our next event tomorrow, October the 6th at 1 p.m. Central European Summer Time, where we will have episode 5 of this first series, entitled From Smallpox to COVID-19, Vaccine Development and Innovation. And the event will feature a special interview segment with Ronald Pierre-Vincenti, of the Chief Executive of the United States Pharmacopoeia, USP. USP is an observer organisation of FIP, and we work closely together in partnership on many synergistic projects and programs. This should be a great interview, and Giovanni will be leading that interview. And I will be leading a discussion after that with Ross and Giovanni to identify key takeaways and to really identify how FIP can support the transformation of vaccination through the sciences, and how we can support not only our pharmaceutical sciences member organisations, but all our members. Colleagues, we'll take your questions from today over into tomorrow's live event, but we would welcome your input, engagement and involvement tomorrow as well. Thanks today to Ross for his superb interviewing skills, and to Colin and Nikolai for their time and all of the valuable insights. What a session. I really compel everyone to view this, and we'll be doing a lot of promotion on this because I think many of the insights we saw today will be so important for us this year and beyond. Thank you all for your attention and your attendance, and we will see you all soon.